And with that, Dr. Reza Seyedsadjadi is going to be coming up. Dr. Reza will now take us through what you can do next if you're currently not in a trial.
I got the good news that I'm going to speak briefly. I was just thinking about that it gets more and more difficult after all that emotion to give a talk to a group. Humbling experience, and always leave CMTRF thinking that we should do more. We should do more. So Susan talked to me before the meeting. We brainstormed about talking about how can we motivate getting enrolled in the study if we are not suitable for a trial. And I suggested that maybe I could tell you briefly about what happens through CMT clinic, and tell you my take on the things. And also, I think some of it Susan also shares with me. So the first visit, you come to clinic, you get a diagnosis, the genetic testing, all those big words about axonal, demyelinating, counseling, talking about things to improve the gait and function, wearing braces, wow moment, as Ken Cornell liked to say, and go to physical therapy, occupational therapy, come back a year later. So what? Why am I coming back to you? Are you going to do the whole same thing from scratch? Probably not. There will be some troubleshooting here back and forth, but you come back hoping for a treatment, hoping for something new to hear from us. We say, okay, research and community, but where is that going? So we like to talk about that. I'd like to tell you that this is the experience that I have in the meeting. With the first visit, I get excited, do the most of the talking and maybe teaching, humbly if I say so. But the follow-up visits is mostly for us to learn from you. So it is us that we take from you, then you take from us to the patients. So I stole this from Mary, as like 99% of my other slides. I thought that this was a nice cycle of showing that we found so many genes, and then where's the bottleneck here? Maybe there are different places, but the place that I feel most, and I will talk more about it, is somewhere in between. It's not just a clinical trial or an understanding of the disease, probably all of these. But I think it is between the candidate therapies and clinical trials that we have, because we hear a lot about the new compounds, this comes, that goes, but that's the part that we have difficulty getting everybody engaged. So I like this example, but this is not the best example of clinical trials. This was about 2 decades ago when they tried ascorbic acid. They showed that in the mouse model, which probably was not the perfect model for CMT1A, that it could help CMT1A, but the human clinical trials didn't prove that, and a lot of money was wasted. And on doing a clinical trial, which was not as fruitful. In fact, some of the studies I look at sometimes that this is the patients on placebo did better than patients on the active arm. Now, I like this review by, again, Mary and the rest of our Inherited Neuropathy Consortium leadership about that the need for finding surrogate points about the study. And this goes back over the past 2 decades about the efforts that INC, through the support that they have got from NIH, from like CMT advocacy groups, have run and patients most importantly. And we learned from you by your participation in these studies and coming and seeing us that what is the rate of disease progression? The way we measured it before and earlier on, was this adequate for us to be able to see changes, detect changes? And was it, in all honesty, helpful to decide or design a trial with enough number of patients to show efficacy of the treatment or screen treatments for. This led to multiple other studies, again, most of them led by INC colleagues and Mary's group, and looking at the natural history of different types of CMT, which are very crucial for developing treatments and therapeutics. And Mary nicely summarized all of the MRI studies with the beautiful pictures. And I stole this slide from my colleague, Dr. Support, to put it in a nice graph that different outcome measures have different level of sensitivity. And if you want to use like more traditional outcome measures on the top corner, you will need like huge number of patients to run a clinical trial to show efficacy of the treatment. But using like more sensitive outcome measures with MRI, for instance, again, Mary's group have been spearheading. You can do trials with very small number of patients. And again, that's thanks to the patient community who have contributed to this study, and also all of us. So why participation matters? I think you already know the answer. So the research is like a micro ecosystem. We all are connected. Clinicians are connected to industry, to advocacy, to patients, to researchers. And if any of these connections is missing, there is not going to be successful. We need to maintain this ecosystem to be able to do the clinical trials, bring the research to clinical trial. And there are far more ways of participation beyond clinical trial. And in fact, most of them are more impactful. So natural history studies, patient registries, genetic testing, Stefan's studies, biobanks, and survey and outpatient reported outcome measures, which is my research interest, and wearable and digital tools. And how does this move the field forward? It accelerates discovery of things that are more meaningful to the disease, clinical endpoints that mean something to the patient clinically. Essentially, what matters most to the patient should be reflected on what outcome measure is trying to measure. Essentially, patient's voice defines the research power. So what we'd like to learn about from you has two different flavors. Types of data that are about the patient's perspective, like the outcome measures. Some of those forms that are exhausting to fill in, like outcome measures and quality of life outcome measures with 36 different questions, functional outcome measures. And then some of them are biomarkers or functional studies, genetics, neurophysiology, imaging, and the tissue. It could be skin biopsy or a blood test. And these are helping us understand the disease mechanism. The common questions that I hear from patients with regards to participation in research or future clinical trials. What if my type is not known? So I used to tell patients that, yeah, 50% of the time you're not going to be able to figure out what type you have. So we heard from Mary's talk that they are more successful in defining or finding the reasons more for type 1 and type 2. But it would be interesting to do more studies, as studies that Stefan is spearheading in terms of defining new genetics. A good example of that would be SWORD, which discovery of it led to relatively rapid drug discovery and the clinical trial. So it would be helpful to try to enroll in the studies to define better genetics. The other things that we heard in the meeting about new compounds and the downstream treatments that would be applicable to more than one type of genetic type of treatment. So this is the study that we are doing in collaboration with Inherited Neuropathy Consortium. So the other common question, again, I think Susan and I were talking about this. Some patients says, I'm mildly affected or I'm very severely affected. How can I still help? And yes, it is very actually important for us to know what extreme of the study you are. So this is like, again, from Mary's group about CMT type 2A, and the patients would have more severe disease. They're going to show us what is the end spectrum of the disease or what's the mild level of disease to define better natural history. Without the extreme ends, we are not going to be able to find a sweet spot to do the clinical trials. So even if you are very severely affected, or you're not affected or mildly affected or asymptomatic, your contribution is going to be very valuable from outcome measure perspective. So what should I do today? I'd say join a registry, share your genetics with us, complete the surveys, donate samples to biobank, and partner with an advocacy group. Every contribution counts. So going back to the first slide is that first visit is, what can my physician do for me? And then the follow-up question is, what can I do for efforts to help everyone else with CMT? And this quote is from Susan, is not from me, but I think it summarizes nicely. So if you don't know what your body is doing today, we won't be able to measure if it's changing the course of the disease. So we are curious about you today, so we can help you tomorrow.